Another example we'll discuss is autism, which is still very controversial, but the preliminary evidence to date suggests that studies of children on the autism spectrum may have different microbes in their gut than the neurotypical children without ASD, often their brothers and sisters. Now, while there is a relationship between these gut microbes and ASD, the causality of this relationship is unclear, and in part the reason for this is that autism often co-occurs with diarrhea and other gut problems, which might themselves be changing the gut microbiome. As a result, it's really difficult to find the controls that you need of children who have the kinds of gut barrier dysfunction that you see in autistic children, but who themselves are neurotypical. So we have to be very careful before drawing any causal or correlative links in this case until we have additional information with more careful controls. However, we can do a lot more in mice than we can do in children. And one fascinating study conducted by Sarkis Mazmanian's group at Caltech involved activating a pregnant female mouse's immune system by simulating a viral attack using double-stranded RNA. Now, when the mothers are under this maternal immune activation scenario, basically a model for RNA viruses, like for example the flu virus, what Sarkis saw was that it led to autism-like symptoms. Led to autism-like symptoms in the offspring. And so what were these symptoms? Well, they included things like repetitive behavior, lack of communication and social interaction, cognitive impairment, and just like in children, gut barrier dysfunction. Now, along with all of these phenotypic defects, the gut microbial community was also altered after maternal immune activation. However, using a probiotic bacterium found in the healthy human gut, Bacteroides fragilis, could actually partially restore some of these issues. So how did this work exactly? Well, profiling by metabolomics, in other words, reading out all the metabolites that were in the gut of these mice, showed that one particular chemical, called 4-EPS, was at much higher abundance in the maternal immune activation mice, and much lower after restoration with the B. fragilis probiotic. What was really fascinating, though, was that administering that one chemical that had been identified, 4-EPS, to otherwise healthy infant mice could cause some of those same autism-like symptoms. So this study is especially important because it suggests a general pipeline for linking microbes, metabolites, and health, including conditions that affect the brain. The first step is to identify altered community profiles, then to find out what metabolites are involved. And then finally, we can test those microbes and or the metabolites that we think are important for specific effects in preclinical models, and then ultimately in the clinic in humans.